We must remember, prior to absorption, all carbohydrate must be converted to monosaccharide form. All carbohydrates must be converted to monosaccharide forms. Okay. Monosaccharide forms can be absorbed directly. No other form of carbohydrate will be absorbed by our intestinal cells. Only monosaccharides will be absorbed. So, be it lactose, maltose, isomaltose, sucrose, or any sort of disaccharide, oligosaccharide or polysaccharide, it must be broken down to monosaccharide form prior to absorption. Now, the enzymes which we have seen or we have studied mostly in the chapter of enzymes were intracellular enzymes. Now, digestion requires extracellular enzymes, means the enzymes which are found outside the cell, What do you call these enzymes which are found outside the cell? Anyone? Enzymes which are found outside the cell are called ectoenzyme. So, all digestive enzymes are ectoenzymes. They are secreted from the cell into the intestinal lumen, where it breaks down the larger molecules. Clear? Any sort of digestion means simplification of larger molecules to their simpler forms. Correct? Now, as you know, the digestion of carbohydrate begins from mouth, the buccal cavity. The buccal cavity is supplied by 3 pairs of salivary glands. Should I name them? Very good. Now, out of which contains maximum amount of fluid and which contains maximum amount of enzyme? Parotid gland contributes maximum of fluid and sublingual contributes maximum amount of enzymes. Okay? Fine. What enzymes do you find here? Salivary amylase. What is the function of salivary amylase? Breaking the carbohydrate to, I tell you what is the taste of glucose? Glucose is cold, it is tasteless. If glucon D jibe dao তাহলে কি হবে? You feel cold, ঠান্ডা, কিন্তু tasteless. So now you tell me, what do you feel after chewing for a certain time a piece of bread or a piece of roti or chapati? Sweet, slightly sweet. So this sweetness is not due to glucose, because salivary amylase cannot break down any polysaccharide up to glucose. It can break down it up to maltose, isomaltose or maltotriose. Clear? Rest of the breakdown takes place beyond the mouth. So then it reaches the stomach, where it is called chyme. Correct? There it gets mixed with what? Gastric juice. pH of gastric juice is highly acidic. Now, the salivary amylase has a pH in basic scale, basic range, that is between 10 and 12. So the bolus, when it reaches the stomach, the surrounding, the outer part of the bolus reaches the pH of gastric juice, that is 1 to 2, but the inside still remains, bolus means a mondo, there is mondo, so inside still remains pH 10 to 12. So the bolus still continue to be digested even though it reaches the stomach for an earlier stage or some time from inside out. From inside out. Then it gets mixed with the gastric juice completely and the salivary amylase completely inhibited. So no more salivary function occurs in the gastric juice. Clear? In the gastric juice, it contains no amylolytic enzyme. Amylolytic enzymes means the enzymes which cleave carbohydrates. So no enzymes is present in gastric juice which breaks down carbohydrate. What next? Then it reaches the duodenum. In the duodenum, it gets mixed with what? CBD, the extracts of the secretion from CBD, right? The bile duct and the pancreatic duct and common hepatic duct. So all sorts of enzymes gets mixed here. Here, due to the high pH of bile, the pH of the medium again changes to alkaline or highly basic. Yeah. Now at this point, pancreatic amylase comes into action. That is the highest potential amylolytic enzyme. It brings down or breaks down all sorts of polysaccharides. Starch, whether it is boiled or unboiled, doesn't matter. Right? Polysaccharide in any form. If there is any glycosidic linkage which has alpha 1,4 glycosidic linkage, then it cleaves. I repeat, the pancreatic amylase as well as salivary amylase both function on alpha 1,4 glycosidic linkage. Do we have any amylase which cleave the other forms of glycosidic bonds? Yes, we do. The very common is beta amylase. But this beta amylase, which works on beta 1,4 glycosidic linkage, is not present in human body. Not only human, it is not present in any primates, any mammals. Then where do you find them? We find them in the secretion of bacteria. The bacteria which can cleave cellulose, because cellulose contain beta 1,4 glycosidic linkage. Now, do we consume cellulose? Yes, we consume cellulose in the form of vegetables and fruits. Correct? But we cannot digest cellulose. Can a cow digest it? How? Do you think the cow is not a mammal? Recall, I said mammals don't have beta amylase. Do you think cow is not a mammal? Bolo. Huh? Anyone? Is there anyone who has already operated his appendix out? কারো অ্যাপেন্ডিক্টমি সার্জারি হয়েছে এখানে? এই কাছে কার হয়েছে? তোর হয়েছে? দাঁড়া। So tell me what you have lost. What is the function of that particular part, what you have lost, what you don't have anymore? Are you having any problem for not having that appendix?
As of now, no.
Then when could you have the problem? In what age?
At that point of time.
What point of time? At the age of 60, 65, 70, or beyond that? After death? After death, will you have any problem? No. Before death, you will have any problem?
After the surgery, I face some
Pain.
yes.
After the pain is gone, now you are having any problem in digesting food? Yes. Then you must be very close to cow. Not all of us. So cows and us, we are different in that particular part of the intestine, which is called cecum, and we call it appendix. The cow and any other ruminating animals. Ruminating animal means jawor kate, who can regurgitate and chew and again swallow for digestion. Why? Because unlike starch, the cellulose produce very less amount of energy. It is energy production wise very less efficient. We have to take 100 grams of rice to survive, to run. A cow has to take 100 kgs of grass to do the same. Correct? Because grass produces less amount of energy. Because cow being mammal, they cannot digest grass, but they still eat grass. Why? Because they have a specialized bacteria in their cecum. The food gets stored for some time temporarily in the cecum, where it is mixed with the bacteria, and the bacteria then chews up all the present cellulose. A very few amount of, so less than 10% glucose is produced. And that is again goes back to the main intestine, small intestine, and gets absorbed. And rest undigested part comes out as a stool. So they contain a lot of fibers, undigested fibers. These herbivorous animals, cow, buffalo, elephant, name some others, zebra, giraffe, their dung is full of undigested these fibers. Our dung also contains the same, undigested. So if we take more and more fibers, it increases the bulk of the stool and it reduces the risk of constipation. Now what happens when a cow is treated with antibiotics? It is dangerous. The cow might die. The cow might die. Why? Because they cannot survive on grass anymore. They have to be given some other starchy food, like rice, like chana, grams, all those. Because it contains starch and protein that they can digest in the small intestine. But they cannot digest grass anymore. Because if all the bacteria in the cecum dies, then they cannot digest cellulose anymore. Okay? Since they consume a huge amount of fibers, they also produce huge amount of gas called flatulence. Do we fart? Yes, all of us, every day, we fart knowingly or unknowingly at least for 4 to 5 times. After taking food, on an average, after 2.5 hours to 4 hours, every one of us will fart depending on the type of food we have consumed. Similarly, the cow also fart. And they fart a lot because they consume more fibers. The fibers remains more time in the intestine, it gets digested, gets fermented by bacteria, it produces more carbon dioxide as well as H2S. If H2S portion is more, then we come to know, the neighbors will come to know. If H2S portion is less, then only you come to know. Right? Fine. Now, next slide. Now, so here in some books might have written the cow have beta amylase, that is wrong. is completely wrong. Genetically, no cow, be it Hindu or Muslim cow or Jewish cow or cow from Palestine, Gaza Strip, no cow, or from Africa, no cow. Arabs, they don't have cow anyway. There is no cow in Arab. Do you know that? Do you know that? In Middle East, there is no cow. Okay. So these cows, no cow has beta amylase. Do we have other amylases? Yes, we have other amylases, but those are found in specialized bacteria and synthetic enzymes. They are used in some treatment. Okay, medicine part. Now, after this part is done, see, it is broken down into amylase, is broken into smaller parts, stretches of carbohydrates called dextrins, limit dextrins. Dextrin is bigger than limit dextrin. When you digest dextrin, it becomes limit dextrins. Okay, when you digest limit dextrin, it becomes maltotriose. When you digest maltotriose, it becomes maltose. Up to here, amylase will function very well, but not till the end. Up to disaccharide form, amylase will bring it down. From disaccharide onwards, we require specialized enzymes secreted by the intestinal cells, intestinal glands. Now, what is so special in these intestinal enzymes? Remember, these carbohydrates, when they exist in monosaccharide form, they exert huge amount of pressure on the cell membrane. This is called osmotic pressure. So, whichever side they will reside on the cell membrane, they will bring water to that side. Okay? If glucose comes more in the intestine and remain unabsorbed, then it will create liquid stool called diarrhea. Right? So, it is not desired. So, what happens? For digestion of disaccharides, nature has given us or taken up a policy like this. Suppose these are the finger-like projections of villi called microvilli. Microvilli. The enzymes, the lactose, say for example. Lactose means glucose plus galactose, right? Like this is there. So enzymes, they cleave here. Then glucose and galactose are separately absorbed. They do that, this digestion, very near to that membrane of microvilli. Why? Because if it goes away here, then what will happen? It breaks down, glucose and galactose will separate. What will happen? It will draw water in a lumen and will cause diarrhea. So, in order to avoid such circumstances, nature has provided us very action of these maltase, sucrase, lactase, isomaltase, these enzymes, very close vicinity towards the microvilli. Okay? So, always keep in mind these things. Rest of things are very easy, you have already studied. Correct? There is nothing much to say. Only thing is, you must remember that we do not have beta amylase, but we do require cellulose for better health. Cows even don't have, but they have extended cecum where they store the food for some time and get digested, but produces less amount of energy, less efficient process of energy production. And these enzymes, which are secreted by the intestinal glands, they exist in very close vicinity to the microvilli membrane. Now, these are the processes how these things are broken down. See, this is salivary amylase, how it broken down, limit dextrin, maltodextrin, maltose, isomaltose. So the sweet taste is due to these, not due to glucose. Now, a few important points about chloride. The amylase. The alpha amylase is activated by chloride ion. Remember, alpha amylase is activated by chloride ion. That's why a pinch of salt helps in digestion of carbohydrate more. We like to eat rice, but only plain rice, but we need only a pinch of salt. That is for two reasons. One, chloride for activation of amylase, another sodium for absorption of glucose. That we will see later. Right? Now, how do you prove that amylase require chloride for activity? How do you prove? Suppose we ask you to design an experiment to prove that amylase require chloride for activity. How do you prove that? Anyone? First of all, you have to collect amylase. How do you collect? Kill a person, cut his pancreas out, then squeeze it. Or how? Ask him to take the smell of glacial acetic acid and then ask him to collect his saliva. Right? Take the saliva, centrifuge it, remove the particulate matters, take the clear saliva and then put it into a parchment pouch. Parchment paper pouch are available, you can put it. Otherwise, you can take the air sac of any fish, function same, whichever is feasible to you, conducive to you, and then place it in a water free from any salt. So what will happen after some time? All the chloride from inside the saliva will come out of the parchment paper. Right? The inside the saliva will contain no chloride. Then it will take overnight, say around 4 to 6 hours. Then next day morning, that saliva you add in a solution of starch, 1 or 2 drops, and then add 1 drop of iodine solution. So the solution will become dark blue instantly. After keeping for some time, the color will not change. That means the salivary amylase is unable to digest or cleave which one? Starch, no color change. Now you add a pinch of sodium chloride or potassium chloride, whatever it is. It must contain chloride. Add a pinch of chloride, mix it and wait for some time. See, slowly the color will disappear. That means starch has been broken down into dextrins or limit dextrins or maltodextrins. That means this experiment proves that addition of chloride renders amylase into active form. Fine. So these are the experiments which can be asked here. These already I have explained. You must remember the names and their sources. Now, this is absorption. Now, this one you have to remember. This will come. And this you have to remember for even your medicine practice also. Forever. These are the lists of glucose transporters present in our body. out of this list, you don't have to remember all. Few I will mark that you have to remember, and what all points you remember, that also I will tell you. So please write down and make your note compatible for examination. Now here, the first column is the isoform, name of the transporter. The second column, the tissue localization, means where it is found, in which tissue it is found, location. The third column is whether it is insulin sensitive, question mark. Yes or no, either of these answer will be there, right? And then what type of sugar it transports. That means preferred sugar for transport. Preferred sugar for transport. These two are not required. So how many you remember? 1, 2, 3, 4. Correct? 4 columns. And number of rows? 6. I will come to that. Okay? First one is GLUT1. First one is GLUT1, G-L-U-T, stands for glucose transporter, GLUT1. Where do you find them? Everywhere. Just remember, everywhere, in all tissues. You find GLUT1 in all tissues. Okay. What do you call that this thing? It is present in all tissues. Ubiquitous. The single word tells everything. Ubiquitous. It is found in everywhere. Ubiquitous. Correct? GLUT1. What does forget about this. Come back to GLUT2. GLUT2. Where do you find them? See, liver, pancreas, intestine, kidney. What is not present here? Brain. Correct? And RBC. Liver, pancreas, intestine means all sorts of intestinal region, abdomen and kidney. All organs of abdomen, GLUT2. Correct? GLUT3, only in brain. GLUT4, above diaphragm, heart, brain, muscle, and what and bat? What is what and bat? White adipose tissue, brown adipose tissue. Clear? This you remember. So, GLUT4, heart, muscle, adipose tissue, brain. So, what is new in it? Adipose tissue. What is new in it? Adipose tissue. First time it is coming, na? GLUT5. See, what is this? That is important to remember for GLUT5. Liver and kidney will have, doesn't matter. But new organ, testis. Right? Testis, presence of GLUT5 in testis is very, very important. We'll see why. Then 6, 7, 8, 9 not required. Nothing you write. Don't write. Just next you write S-GLUT. S-G-L-U-T. Up to 5 you write, then S-G-L-U-T. Okay. Now it has two forms, 1 and 2. It has two forms, 1 and 2. Is GLUT1 present in two see, everyone will tell you what to read, but very few will tell you what not to read. Huh? So this is something which you must read. You must remember the first 5 of the table, then the 6th and 7th under the same heading called SGLUT. It has 2 forms, SGLUT1, SGLUT2. Clear? So up to the second column we have remembered, right? Now your table is ready up to second column. Huh? Come to third column. What is the heading? Whether they are insulin sensitive or not. Just write yes for GLUT4 and no for rest all. Clear? Yes for GLUT4 and no for rest all. That means except GLUT4, no other glucose transporter is insulin dependent. Now where do you find GLUT4? In which organ? What is a specialized organ I said? Muscle and adipose tissue. So muscle and adipose tissue require insulin for absorption of glucose. So when you have diabetes, that means insulin deficiency, then which tissue will get affected? Muscle and adipose tissue. That's why we lose weight. We become lean. Because muscle and adipose tissue go starving. They don't get the sugar. Although glucose is there in blood, plenty of glucose is there, but they cannot utilize it because of lack of insulin. But nothing goes wrong with erythrocyte, brain and kidney and intestine. Why? Because they contain other transporters which are insulin insensitive. Clear? That is a big point you must remember. Muscle and now find out where did I mention heart in tissue. Which transporter? GLUT4 and correct? So heart muscle don't get upset. Clear? Heart muscle is different from skeletal muscle. Remember, GLUT4 is mostly present skeletal muscle. Heart muscle can survive with GLUT1. Clear? Fine. Now, what is the next column? All glucose transporter must transport glucose? Not necessarily. They might have preferences. Right? So what are the preferences? So GLUT1, what is the function of GLUT1? We will see later on the previous diagram. Their major transported sugar is glucose and glucose only. Glucose and glucose only. GLUT2, what is there? Glucose and fructose. But glucose is low affinity. Glucose is low affinity. That means it transports glucose, but when the glucose concentration is very high, then only it transports across the membrane. Now, find out where do you find these? Liver. Now, what is the function of liver? To store excess of carbohydrate. So unless the blood is full of, blood contains plenty of glucose, it will not absorb glucose, so that other tissues can get the glucose. Then only if it is excess, then liver will take it for future use, otherwise not. So that's why GLUT2 is present in liver. Fine. GLUT3, glucose high affinity. It transports glucose only with high affinity. Means what? Very low concentration. Why do you find GLUT3? Yes. So brain never goes starving. Very small amount of glucose is there, it will take up. But liver, just opposite. It will wait until the glucose concentration is very high. Clear? GLUT4, glucose high affinity. Correct? GLUT5, what is the first? First is fructose, not glucose. They prefer transport of fructose. They prefer transport of pentose sugar, xyloses, riboses. GLUT5 prefer fructose, then the other pentoses. So all sorts of pentose absorption is done by GLUT5. Where do you find fructose? In which body fluid? Semen. So the testes require GLUT5 to maintain high concentration of fructose in the testicular fluid or semen. Clear? Now you come to SGLUT. The SGLUT name is sodium dependent glucose transporter. The sixth one, what you have written. Glucose transporter. It is 1 and 2. Two subclasses are there. The subclass 1 is present everywhere in all intestine. Subclass 2 is present in renal tubule. They are insulin independent. They are sodium dependent. Clear? They function only on glucose and glucose only. No other sugar they transport. But they require sodium. That is why when glucose is given to a starving person to break his fast, it is always given along with a pinch of salt. Otherwise, it will not be absorbed. Clear? Next, write down, there are some drugs which are used to block this one. Dapagliflozin. This drug is used for SGLT2 inhibitor. SGLT2 inhibitor. Why? What is the function of renal tubule SGLT2? SGLT2 actually reabsorbs the glucose, which is filtered during the formation of urine, but again it is reabsorbed. Correct? In absence of SGLT2, the sugar or glucose will appear in urine because it is not reabsorbed. Correct? So a person who is having diabetes, having more glucose in the body, blood, can get rid of it if we give them this drug. So what will happen? The urine will get a lot of glucose. Glucose 2 plus, 3 plus. Correct? Although the blood glucose level might be 120, 160, below the renal threshold. So a person taking these drugs may come to you with blood sugar or blood glucose below renal threshold, but still sugar is more in the urine. This could be one reason, that urine shows presence of glucose, or sugar, but blood glucose level is below 180 milligram per deciliter. So this is renal threshold for glucose. How do you explain this? Number one, the person might be taking specific SGLT2 inhibitor, dapagliflozin. Correct? This could be one answer. Another answer is, in certain patients who are chronic diabetic patient. No, this answer only. This answer only sufficient. So, if you get this kind of questions in exam, then this should be your answer. The person might be taking SGLT2 inhibitors. Okay, this comes in the examination. Important. Fine. So remember this, and that's all for absorption of carbohydrate. Rest all are not required for examination purposes, whatever it is. Any question so far? See, books have given everything or many things. YouTube channels, plenty of YouTube channels are there explaining, speaking more vividly than I can speak, with better English, with better Bangla, better Hindi, whatever language you prefer, Kannada, Telugu. But still, the classroom teaching is something where you can interact with the teacher. So I give you the chance to interact with me regarding subject. Do you have any question, Amrita Chakraborty? Or are you still angry with me because I told you to remove your mask? You have such a beautiful face. Why you are masking with mask? Good. Go ahead. Any question? No question. Hey, first bench. One question? Ball. Huh? SGL2 টা SGL2 এরা সবাই কি করে, সেলের ভিতরে ঢোকায় গ্লুকোজকে। SGL2 ঠিক উল্টোই থাকে, মানে যে গ্লুকোজটা বেরিয়ে যাচ্ছিল সেটাকে আবার সেলের মধ্যে ঢুকিয়ে দেয়। রেনাল টিউবুলে যখন ফিল্টার হয় তখন ইউরিন তৈরি করার সময় তখন গ্লুকোজটা ফিল্টার হয়ে বেরিয়ে যায়, সাইজ ছোট বলে, তখন সেই গ্লুকোজটাকে আবার রিঅ্যাবজর্ব করে নেয়। নর্মালি কি আমরা ইউরিনে গ্লুকোজ পাই? পাই না। পুরো ইউরিন পড়েছিস, পাই না। তাহলে ইউরিনে গ্লুকোজ পাই না কেন? বিকজ অফ অ্যাক্টিভ SGL2। আমরা কি করি, ড্রাগ দিয়ে এটাকে বন্ধ করি, বুঝলিস? SGL2 ইনহিবিটর দিয়ে বন্ধ করলে কি হবে? ওটা অ্যাবজর্ব হবে না, তাহলে সুগারটা, এক্সেস সুগারটা ওখান থেকে বেরিয়ে যাবে। ডায়াবেটিসে কি হয়, এক্সেস সুগার যদি শরীরে জমা হয় তাহলে সেক্ষেত্রে নানা রকম ডিজিজ হয়, তাই তো? পেরিফেরাল নিউরোপ্যাথি, রেটিনোপ্যাথি, নেফ্রোপ্যাথি, এগুলি হবে না, তার রিস্কটা কমে যাবে। মজার ব্যাপার হচ্ছে, এটা দিলে কি হাইপোগ্লাইসেমিয়া হবে? হবে না। কেন হবে না? কারণ ওই গ্লুকোজটা তো অলরেডি আমাদের শরীর থেকে সাবস্ট্রাক্টেড হয়ে গেছে, বেরিয়ে গেছে। তাহলে হাইপোগ্লাইসেমিয়া যদি তখনই হতো, যদি হওয়ার ছিল, তাহলে যেই মুহূর্তে ইউরিন প্রোডাকশন হলো সঙ্গে সঙ্গে আমার হাইপোগ্লাইসেমিয়া হতে পারতো, তা তো হয়নি। তার মানে ওই ইউরিনটা আমার ব্লাড থেকে বেরিয়ে যখন ইউরিনে চলে যাচ্ছে সুগারটা, তখন তো আমার হাইপোগ্লাইসেমিয়া হয়নি। তাহলে SGL2 ইনহিবিটর দিলে হাইপোগ্লাইসেমিয়া হবে কেন? বলতে পারলাম? আর কোনো প্রশ্ন আছে? বলতে পারলাম? পারলাম না? পারলে? আচ্ছা, আর প্রশ্ন আছে? Anyone else? No. আচ্ছা, কটা বাজে? আরে খুব তাড়াতাড়ি হয়ে গেল। একটু চেঞ্জ করো। আজকে পড়াবো? পড়ি যাও, হ্যাঁ, বন্ধ করে আবার মার, এসকেপ মার, বন্ধ করাটাকে। ওই মাউস দিয়ে ক্রস করার পরে, হ্যাঁ দাঁড়া, আমি বলে দিচ্ছি। এটা কর, ওই পাশেরটা, এটা ছোট্ট একটু শুরু করে ছেড়ে দিচ্ছি। তাই না? শুরু করে ছেড়ে দিচ্ছি, আবার 7.5 আমারই মুখ দেখতে হবে। ওই নিচেটা মার, F5 মার, F5 মার, F5 কিবোর্ডে F5 মার, হ্যাঁ তাহলেই হবে। গ্লাইকোলিসিস, হোয়াট ইজ গ্লাইকোলিসিস? এর থেকে সহজ কোশ্চেন হয় না, তুই বল। ব্রেকডাউন অফ গ্লুকোজ, আচ্ছা তাদেরকে ডাইজেশন বলে। এক্ষুনি পড়লি, বলে কি ব্রেকডাউন গ্লুকোজ ডাইজেশন বলে? বল বল বল। অক্সিজেন অফ গ্লুকোজ বল, তাই না? গ্লুকোজকে ভাঙলে কি হবে? ভাঙা যায় না, গ্লুকোজ অক্সিজেন অফ গ্লুকোজ। তারপরে কোথায় হয়? আচ্ছা বসো। So glycolysis is the oldest metabolic pathway operating in cytosol of all living cells. তাই তো? এর অনেকগুলো নাম আছে। কি কি নাম আছে? একটা নাম হচ্ছে এমডেন মেয়ারহফ ফার্নেস পাথওয়ে, এমডেন পাথওয়ে ও খালো বলে, তাই না? আচ্ছা, কোথায় হয়? লোকেশন হচ্ছে সাইটোসল। আমি বললাম সাইটোসল আর সাইটোপ্লাজম কি পার্থক্য? হোয়াট ইজ ডিফারেন্স? বলো অমৃত চক্রবর্তী, what is the difference between cytosol and cytoplasm? সাইটোপ্লাজমে অনেক সেল অর্গানেল থাকে, তাকে সাইটোসল বলে। মানে সাইটোসল প্লাস সেল অর্গানেল ইজ ইকুয়াল টু সাইটোপ্লাজম। দ্যাটস হোয়াট ইউ ওয়ান্ট টু সে? এনিওয়ান এলস? Cytoplasm minus cell organelle minus cytoskeleton is equal to cytosol. Cytosol is a liquid part where only dissolved enzymes remain. Sit down. So glycolysis is a process by which glucose is oxidized, but it is oxidized, but in no steps of glycolysis oxygen is required. Okay? So where do you find oxygen? Where actually oxygen is used? In electron transport chain, if you can recall. Okay? Glucose glycolytic pathway does not require oxygen, but it depends on which pathway it chooses. Later on that it can be named either oxidative or aerobic or anaerobic glycolysis. So glycolysis can be of two types, aerobic and anaerobic. What is aerobic glycolysis? The end product of aerobic glycolysis is pyruvate. What is anaerobic glycolysis? The end product of anaerobic glycolysis is lactate. Now we come, what is the importance of anaerobic glycolysis? Why do we need anaerobic glycolysis and why do we have anaerobic glycolysis? Can anyone tell me? The site for anaerobic glycolysis is the tissues which contain no mitochondria. Am I right? Mitochondria is required for electron transport chain, which requires oxygen, correct? Now, if there is no mitochondria, then there will be no aerobic glycolysis. True? Huh? But there is a tissue which contains plenty of mitochondria, but still that tissue operates anaerobic glycolysis. So give me an example, the tissue which has no mitochondria, that's why he cannot operate. দেখে নে, কাকে দেখছিস? কাকে দেখছিস? তোকেই জিজ্ঞেস করছি, দেখে নে, প্রাণ ভরে দেখে নে। So give me an example of tissue which cannot operate oxidative or aerobic glycolysis due to absence of mitochondria. এমন একটা টিস্যুর নাম করো যেটা মাইটোকন্ড্রিয়া নেই বলে অ্যারোবিক গ্লাইকোলিসিস করতে পারে না। Mature RBC. Mature RBC. Now, name a tissue which has plenty of mitochondria but still operates anaerobic glycolysis. Still operates anaerobic glycolysis. Skeletal muscle. Why? Because anaerobic glycolysis produces energy instantly. Suppose you are sitting here and suddenly you find something is very cold wrapping around your leg. What will happen? Suddenly you will jump. Before that moment, before that very moment, did you know or did your muscle know that you are going to jump? No. The instant movement of muscle require instant burst of energy, which is provided by anaerobic glycolysis. Why not aerobic glycolysis? Because anaerobic glycolysis produces less energy. Aerobic glycolysis produces more energy, but aerobic glycolysis takes more time. Anaerobic glycolysis takes less time. Anaerobic glycolysis produces lactic acid, which accumulates in the muscle, then it causes muscle cramp. I ask you to climb up to a 5-story building 5 times. What will happen? After 2 times, you will stop automatically. Why? Due to muscle cramping. Why? Muscle has accumulated a lot of lactic acid. But ask the same to Virat Kohli. He will finish the job. Why? Is he more fit? Yes, because he practices every day. More you practice, more you get used to how to cope up with high content of lactic acid accumulation in the muscle. Correct? Next. This is the net reaction of glycolysis. 1 molecule of glucose broken into 2 molecules of pyruvic acid in presence of 2 molecules of NAD plus 2PI, which gets converted to 2 NADH, and 2 molecules of ADP gets converted to 2 molecules of ATP. So glycolysis is a site of substrate level phosphorylation. There are 2 types of phosphorylation. One is oxidative phosphorylation, another is substrate level phosphorylation. Any Tom, Dick and Harry, any cell can operate glycolysis, can produce small amount of energy by substrate level phosphorylation, by production of ADP. But only the cells which has mitochondria in it can produce more amount of energy more efficiently by oxidative phosphorylation. Clear?